All right, so this is linkage and mapping part 2. We're just going to add a few things and then show what it looks like when we draw crossing over with chromosomes. All right, so we know this whole, everything comes down to this kind of a cross. Take a dihybrid, this is a 2-point cross, also called a 2-point mapping cross, and we get 4 types of things out of it. So these would be gametes, but these can also be represent the phenotypes for whatever we're looking at. And then maybe we'll do a couple of problems. But and so we saw before that whichever of these categories is the most are the parentals. So let's say we had an example like this. We saw 10% were this, 10% were this. That's 80%, and then 40 and 40. So you look at that, and you say these are the parentals. These are the recombinants. The recombinants are 20%, and so the map units are 20%. So that part doesn't change. But what changes is the thing that is the parentals in this case are big A and little b, and little a and big B. And so we're used to seeing big A and big B and little a and little b be the parentals. And so that brings up this idea of coupling and repulsion. And so this is what we see when big A and little b are linked together. That's called repulsion. And so that means we have to change the way we represent this thing here. Hang on one second. All right, leave it. Don't cause any trouble. So what does that mean? So let's take a look. So we're going to use a different sort of notation. So this just means that we're heterozygous for big A and little a, big B and little b. It doesn't tell us anything about whether the genes are linked or not. So if the genes are linked, we draw them like this. And this suggests or indicates that they're on the same chromosome. The book will often use this to represent the same thing. And that kind of makes sense because this is on one chromosome, one homologous chromosome, and that's on another. But in practice, we just draw one line. And this is what, if the two capital letters are together, or in other words, if the two dominant alleles are together and the two recessive alleles are together, we call this the coupled configuration. But we can also draw it like this. It's possible that the big A goes with the little b starting out, and the little a goes with the big B. And we call this repulsion. This is just old terminology that means that sort of the way it was originally described is that the dominant alleles tend to go away from each other. And over here in coupled, the dominant alleles tend to stay together. So if we do a test cross with these guys, we would cross it to that, which is a test cross. Just another way to write that as being homozygous for both. And then, so because A and B are together at the beginning, they tend to stay together. So that means if it's coupled, then these will be your parentals. But only when you get crossing over, and the way we'll draw crossing over is an X right here. So now big A comes down and goes with little b, and little a comes up and goes with big B. And so these are your recombinants. So if you could have the same genes and the same distance, but depending on how you do the cross, the things that are your parentals will be different. So in the last example, we used 20%. So that means these are recombinants, and these are the parentals. And that's what makes sense. We're used to seeing big A and big B together, so that makes sense. But if we use the same genes here, now big A and little b are going to tend to stay together, and little a and big B are going to tend to stay together because they started out together. And then down here, we could have little a and little b, big A and big B. Now these will be our parentals. These will be our recombinants. And because these are the recombinants now, they'll be 10, and these guys will be 40. Now, if we see the cross and we're just doing genetic distance, we don't really care if it's in coupled or repulsion. If we see these numbers, we know that the distance is 20%, just because the lower numbers are the recombinants no matter what. But in this case, the recombinants just have a different sort of genotype. So where do these coupled and repulsion things come from? Well, remember when we said we could, so we're doing a dihybrid cross. So that means we have this, and we do a test cross, or not a dihybrid cross, but we have a dihybrid, and we do a test cross. But this dihybrid, can come from two different ways. So remember, the parents can look like this, and that will give us that. Well, if these genes are linked, this means in this parent, the big A and the big B start out together on the same chromosome. And over here, little a and little b start out together on the same chromosome. So what we can rewrite this, it gets big A and big B from one parent, and it gets little a and little b from the other parent. So that's what we mean by coupled. And then we do this test cross, and then big A and big B and little b are the parentals, and then big A and little b and little a and big B are the recombinants. Remember we talked about recombining alleles? So we start out in the parents with big A and big B together and little a and little b together. And now we've shuffled them through meiosis, but now instead of doing independent assortment, we're doing crossing over. Another way to get that dihybrid is to start with something like this. So now in this parent, big A and little b start together. And in this parent, so this is parental generation, so these guys start out like this. So then the dihybrid, if we wrote it without thinking about linkage relationships, we would just write it like that. But now that we know these genes are linked like this, we get one chromosome from this parent and one chromosome from this parent. And now, when we do a test cross, big A and little b tend to stay together. Little a and big B tend to stay together. And then when we get a recombination, big A and big B and little a and little b. So these guys are now the recombinants, and these guys are the parentals, because that's what the parents look like. Big A and little b were the parents. You might be tempted to say that these are the parents in this generation, but this is the F1. So let's look at some chromosomes. What does this look like in a chromosome? So the way we draw this in a chromosome is we have to remember that chromosomes line up during meiosis like this. Meiosis 1. And we were all worried before about drawing different pairs of chromosomes like this. But now we want to think about them, and sister chromatids always have to start out being identical all the time, 100% of the time. Big A and little a on the sister chromatids doesn't make any sense. And then little a goes on this one. But now we're going to talk about a linked gene. And so maybe a B is here and a little b is here. Now the good news is we don't really have to worry about this chromosome because these genes are linked on the same chromosome. But it's a pain in the neck drawing chromosomes like that and trying to draw how they cross over and stuff. So we're going to take these chromosomes and we're going to turn them sideways. And I'm just going to make them into a line. So now this is going to be chromosomes during meiosis, and they're going to look like this. So that's just taking this thing and turning it sideways and then not worrying about not making them kind of loopy like that. And then we'll put a centromere in there somewhere. And so now we have these are sister chromatids. So what does that mean? It means they're going to be identical. These are sisters. They're going to be identical. After crossing over, they might be different, but when they start out, they're going to be the same. So I would have put the genes in slightly different order just because it's easier that way. So let's say this is coupled configuration and big. So what we know is that it's going to be heterozygous for two genes, and more than that, we're going to say that it's in coupled configuration. So it's going to look like that. So that means during meiosis, we have this top homologous chromosome or homolog with its identical sister chromatids. It's going to look like that. And this is going to look like this. So that's how we're going to draw chromosomes. Draw them sideways. Always put an allele in each of the appropriate positions and always have the sister chromatids be identical. And we can this is coupled. We can draw them. We'll draw them in repulsion in a minute. So this is what one set of chromosomes. So like if we looked over here, we did meiosis. Eventually, we would get these kinds of gametes. And because these genes are linked, big A and big B will be together, little a and little b will be together. And so here, if we don't get any crossing over, we'll get only big A and big B. When crossing over happens, now pay attention, because crossing over happens between homologous chromosomes. So that's like happening between here. It doesn't happen between, a lot of times students will draw this like this and say, there's my crossover. That's not where it goes. So the crossover always has to happen between chromatids of homologous chromosomes, not between sister chromatids. So what happens when we get a crossover? Well, what happens is this chromosome is here, and then it breaks. And then we'll say, let's make these chromosomes different colors. So we'll make this chromosome red, and we'll make this chromosome blue. When we get a crossover, this red chromosome, so this top red chromosome stays the same. So let's put this up here. So that doesn't have any change. So that looks like this. Then this other one looks like this. But what happens then is it breaks, and this blue part comes up and connects up here. So now our sister chromatids are not identical, but that's because of crossing over in this case. And now they look like this. In our centimeters down here somewhere. And then this bottom chromatid starts out blue, and then it breaks and connects with this red one up here. And then the bottom one stays blue. And so this has a little a on it. But now because it broke and connected with that red one, it has a big B on it, and the little one stays the same. So that's what sort of happens during meiosis 1. And then they separate a couple times, and we end up with these kinds of gametes. The ones that don't undergo crossing over are parental gametes, and the ones that do undergo crossing over are recombinant gametes. So whenever there's a crossover, you end up with 50% parental gametes and 50% recombinant gametes. So where this idea of genetic mapping and frequency of recombination comes along is that recombination only happens sometimes. Well, it only happens between these two genes sometimes. So in another meiosis, another cell, we start out looking like that with our identical sister chromatids. Got some centromeres here. No crossing over. So this is with what we call a single crossover, or an SCO, where we just have one crossover between the genes. But here we have no crossover, because most of the time you don't have a crossover. There's crossovers happening everywhere, but we're talking about, is there a crossover here between these two genes? If there's no crossover, then we just get these gametes, because we don't have any recombination. Because the genes are linked, we need a crossover in order to generate recombinant gametes. So these are all parentals. So so we have meiosis is happening like a million times, and so on average, most of the time there's no crossover, so we get mostly parentals. Some of the times there is crossing over, and then we'll get some recombinants. And so, depending on how many recombinants we get, that tells us the genetic distance. By genetic distance, we mean how close they are together this way. If they're super far apart, that means there's a lot of room in between those two genes for a crossover to happen. So it's more likely for a crossover to happen. The more often a crossover happens, the more recombinants you get. And the more recombinants you get, that higher recombination frequency, and that tells you the distance. If they're really, really, really close together, so let's look at another page. Let's say we got some genes that are really close together. Now, normally in a classroom setting, let's put a gene here. And we'll put a gene down here. Normally what I would have you do is throw erasers at the chalkboard and see how often you can get, if we just, and think of crossing over as being random across here. And wherever we're going to throw an eraser between here, and wherever we hit that with that eraser, that's where a crossover is going to happen. So we hit one there, hit one there. It's going to be easier to get our eraser to hit here, and every now and then we'll get one over here. But the eraser is going to hit over here more because there's more room. And so crossovers happen randomly across here. And if they're randomly, they're just a higher probability that there's going to be a crossover between C and B, because C and B are super far apart. But the probability of getting one between A and C is lower. And so you'll get a lower frequency of recombination between A and C than between A and B or between C and B. And that's how we make genetic maps. And genetic maps are super important. We haven't talked too much about that, but maybe we can talk about that next. All right. So one more thing we're going to add to this is what's this deal with? So I said the maximum frequency of recombination is 50%. So remember, we said that if recombination percent equals 50, the genes are unlinked. And if the recombination percent is less than 50, the genes are linked. And if the recombination frequency is greater than 50, we said that never happens. So why? So if genes are close together, you don't get crossing over very often. I think I drew those a little too big. Come on, eraser, do your thing. I need a little room there. So let's pretend that these genes are really far apart. That means we're going to start to get a lot of crossing over between there. So if we get one crossover between here, like we just showed, we'll get big A and big B together, That's a parental. With big A, we'll cross over and go with little b, and that'll be a recombinant. Little a will cross over and give you big B, that'll be a recombinant. And then big A and little a and little b at the bottom will stay together, that'll be a parental. What happens when the genes are really far, so let's, I'm going to redraw this so I can show you both things. So I just want to draw it twice. So you should get some practice drawing this. Always have your sister chromatids start out identical. Homologous chromosomes aren't identical. I'm going to put these in here to make it easier. Just fit them in. So if we get no crossing over, big A and big B stay together, big A, big B stay together, little b stay together, little a, little b stay together, we get all parentals. If we get a single crossover, then we get this parental, and then this recombinant. Big A goes with little b, so that's this. And then little a goes with big B, and then little a goes with little b. So even if crossing over happens 100% of the time, let's say every single time we do meiosis, we get a crossover, then we still are only going to get 50-50 parentals and recombinants. So that's why this can't go over 50%. Because even if we have crossing over happen 100% of the time, the maximum number of recombinants we can get is 50%. Then another thing that messes with recombination is when the genes get really far apart, we get a crossover here. If they're far apart, it's likely that that crossover will happen again. When that happens, big A and big B up here will stay together and give us a parental. But now what happens, let's make this colored again. So we'll make this red and we'll make this blue. So what's happening in this case is the red cruising along here, and then we break the chromosome and it connects to blue. So we got red and it connects to blue down here, but then it breaks again and connects back up to red. And the reciprocal thing happens on the bottom, where red will be in the middle and blue will be here. And so what happens in this case, if we have an A here, and so our top one up here isn't involved, and our bottom one down here isn't involved, and here's our centromere. So that happened between homologous chromosomes. And our A was here, and our B was here. And our little a was here, and our little b was here. So what happened is we got a big A, and we crossed over and picked up some of this blue chromosome, but there's no letter here, so we can't really tell by just looking at the offspring that this happened, because there's no gene here that we can see. And then it crosses up over here, and it goes back to being a big B. So if we just look at the offspring, big A and big B start together, parental, 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 parental. So even though we have multiple crossovers, we see they're all parental, so they don't really get counted as crossovers. So what that tells us is in those cases where we have a genetic map, let's say we have A and B here, and we get like, say, 30%, but then if we had a C in the middle that was like 20 and 15, that's why it's inaccurate when the genes are really far apart, because double crossovers make it look like the crossover didn't happen. So remember that these numbers come from number of recombinants over the total, and these would not get counted as recombinants. They would be parentals. They would go into the total, but they wouldn't be recombinants. So just to say it more simply, is that when genes get really far apart, the apparent recombination frequency is lower than it should be because double crossovers make it look like no crossover happened at all. And so it makes it look like parentals. I think that's all we need for linkage. So I do have on the course web, a lot of these questions will come up in the homework, but also problems and video solutions to problems and things like that are already on the course web. If there's any of those you need me to clarify for you, just let me know. So we'll leave this one here. Maybe this one will actually be shorter than 45 minutes. And I'll probably come back and do a chi-square one next.